After studying this module, you shall be able to know the importance of DNA in living system, learn the assembly of DNA and its constituents, identify various types of physiological modifications in the assembly of DNA. Deoxyribose nucleic acid or DNA is the primary genetic substance of living organisms and some viruses. It is a major macromolecule that carries information important for every form of life. This information in DNA is transcribed to RNA and then further coded to proteins for performing functions of an organism. Most DNA consists of two biopolymer strands coiled around each other to form a double helix. DNA are polynucleotides formed of two complementary strands that are twisted around each other as double helical structure. The two strands are formed of monomers and are known as nucleotides. Each nucleotide in turn contains one of the four nitrogenous bases, guanine, adenine, thymine or cytosine, and two deoxyribose, which is monosaccharide sugar and a phosphate group. These nucleotides in the double helical structure are placed nearby covalent bonds between OH group of sugar of one nucleotide and the phosphate of the other, subsequently resulting in a sugar-phosphate backbone. Two strands in DNA are placed near each other as per Chargaff's rule. A always pairs with T and C pairs with G with hydrogen bonds. Within living cells, DNA is structured into extended threads like arrangement called chromatin that condenses to form chromosomes during mitosis or meiosis. Prior to cell division, the DNA content of the cell is duplicated by progression of DNA replication in case both daughter cells with its individual whole set of chromosomes. In eukaryotic organisms, DNA is present inside the nucleus and a small proportion in semi-autonomous organelles like mitochondria or chloroplasts. In contrast, prokaryotes store their DNA in the protoplasm. Contained in the cell, DNA is allied with proteins such as histones or polyamines that make the DNA compact and help in higher orders of its organization. These higher order structures called nucleosomes stabilize the DNA and helps in modulating control of synthesis and interaction between DNA, RNA and proteins. The polymeric nature of DNA was understood in the late 1930s, but it is in the year 1953 that Watson and Crick presented working model of DNA by obtaining data from two sources: base composition of hydrolyzed samples of DNA and X-ray diffraction studies. During 1950s, Erwin Chargaff and his colleagues found the composition of DNA from various organisms by using chromatographic methods. The following conclusions were drawn from the data. First, the amount of purine residue is proportional to the amount of pyrimidine residues of DNA of any species. Second, the amount of adenine is proportional to the amount of thymine and of cytosine is equal to guanine. Third, the percentage of A plus T does not necessarily equal the percentage of G plus C. The ratio, although varies greatly among species, is constant for particular species. The quantitative relationship given by Chargaff is known as Chargaff's rule and is universally applicable to all species. X-ray diffraction analysis of DNA was first carried out by William Asbury in 1938 and highlighted the presence of repeating units of 3.4 angstrom in DNA in 1947. Later, It was Franklin and Morris Wilkins in 1950 who showed that DNA produces a characteristic X-ray diffraction pattern and concluded that DNA is a helix with two periodicities along the long axis, one at 0.34 nanometer and other at 3.4 nanometer respectively. DNA is a highly stable molecule and it is therefore suitable for the storage of biological information. The sugar phosphate backbone of DNA is impervious to hydrolysis. Moreover, the two strands of the double helix impart the same genetic information due to their complementarity. Bulk of DNA within eukaryotic cells is non-coding and do not serve a function of encoding proteins. This may amount to approximately 98% in human cells. The two strands of DNA are antiparallel, running in reverse directions with respect to each other, one backbone being 3' and the further 5'. Joined to each sugar is either a purine or a pyrimidine nitrogenous base aligned in a sequence that beside the backbone encrypts biological information. Information from DNA is transcribed to RNA, which further translates it to protein molecules that are true effector molecule of genetic information. Discovery of 3D structure. DNA is a heteropolymer of deoxyribonucleotides. It was primarily identified and taken out by Frederick Mischer. However, the structure and function of DNA remained obscured for several decades. James D. Watson and Francis Crick proposed three-dimensional structure of DNA in 1953 with the following main features. First, the helical DNA chains coiled around a long axis forming a right or left-handed double helix. Second, the two chains are antiparallel. This is that C5' to C3' orientations run in opposite directions. The two chains cannot be separated without unwinding to helix, a phenomena known as plactonymic coiling. Third, the bases of the chains are flat structure arranged perpendicular to the long axis. They are stacked on one another at a distance of 0.34 nanometer. Fourth, the antiparallel chains of DNA are complementary and not identical. The purines and pyrimidines in both the chains are paired to one another by the formation of hydrogen bonds. A pair with T by 2 hydrogen bonds and G with C by 3 hydrogen bonds. Although every discrete repeating unit is very minor, DNA polymers can be very big molecules comprising millions of nucleotides. For example, the biggest human chromosome, chromosome number 1, comprises of around 220 million base pairs and is about 85 nanometer in length. Within the living systems, DNA occurs as a pair of molecules that are placed firmly close by hydrogen bonding between the nitrogenous bases. The strands are entwined as a double helix. Nucleotide repeats comprise together the section of the backbone of the molecule that place the chain close and a nitrogenous base. It networks with the other DNA strand in the helix. A nitrogenous base linked to sugar is known as nucleoside, and a base linked to a sugar and one or more phosphate groups is known as nucleotide. A polymer containing multiple connected nucleotides is known as polynucleotide. The support of each individual DNA strand contains alternate phosphate and 2' deoxyribose sugar residues that are joined by phosphodiester bonds among the 3rd and 5th carbon atoms of nearby sugar rings. This specific bonding pattern makes each strand of DNA double helix directional and asymmetric. As a result, the two asymmetric strands move in reverse direction and are antiparallel. The asymmetric terminals of DNA strands are known as 5' and 3' ends, with the 5' end has a terminal phosphate group and 3' end a terminal hydroxyl group. Double helix structure is primarily stabilized by two forces: hydrogen bonds among nucleotides and base stacking connections between aromatic nitrogenous bases. In the aqueous environment of the cell, the conjugated bonds of nucleotide base arrange vertical to the axis of the DNA molecule. This alignment minimizes their interactions with the solvation shell and consequently the Gibbs free energy, imparting stability to DNA. Nitrogenous bases. The nitrogenous bases, also called nucleobases, are categorized into two forms: the purines and pyrimidines. Purines, adenine and guanine, are merged 5 and 6 membered heterocyclic compounds, and the pyrimidines, cytosine and thymine, are 6 membered rings. A fifth pyrimidine, uracil, is present in position of thymine in RNA and is not found in DNA. Grooves. Identical helical strands that make the DNA backbone have been observed outlining the spaces or grooves among the strands. These spaces are adjacent to the base pairs and offer a binding site for proteins. Since two strands are asymmetrically situated in reference with each other, the furrows are unequal in size. Larger of the two grooves, the major groove, is 22 angstrom in span, while the smaller minor groove is 12 angstrom in width. This means that the edges of the bases are more accessible in the major groove. As a result, proteins such as transcription factors that can join to particular sequences in double-stranded DNA generally forms connections to the edges of the bases open in the major groove. Base pairing. In a DNA double helix, any form of nitrogenous base on one strand bonds with only one form of nitrogenous base on the other strand. This property is known as complementary base pairing. The purines form hydrogen bonds to pyrimidines, with adenine bonding only to thymine by 2 hydrogen bonds and cytosine bonding only to guanine via 3 hydrogen bonds. This set of purine-pyrimidine nucleotides binding together through the double helix is known as base pair. Since GC base pair has one additional hydrogen bond, it is more even in comparison to AT base pair. Consequently, DNA molecules with higher GC content are relatively more stable. The constancy of the double-stranded DNA depends on the GC content, length, as well as on sequence, that is, stacking is sequence specific. The stability of DNA can be known in terms of melting temperature, that is, the temperature at which half of the double-stranded molecules are transformed to a single-stranded molecules. Melting temperature is dependent on the ionic strength and the concentration of DNA. Under laboratory conditions, the strength of contact can be calculated from the temperature essential to break down the hydrogen bonds, their melting temperature, also known as TM value. Long DNA helices with a more GC content has harder interacting strands, while smaller helices with more AT content have weaker interacting strands. Regions of the DNA double helix that are isolated easily, like as the TATAAT or Pribno box in several promoters, inclined to have a high AT content, making the strands easier to separate and thus carry out biological functions such as replication, transcription, DNA repair and regulation. Since hydrogen bonds are not covalent and are weak, they can be fragmented and joined again comparatively easily. The two strands of DNA in a double helix can consequently be drawn apart, such as zipper, either by a mechanical force or high temperature. The property is valuable while assessing functional role of DNA, and as a consequence of this complementarity, all the information in the double-stranded arrangement of a DNA helix is reproduced on every strand, which is important in DNA replication. This reversible and precise interaction between complementary base pairs is important for all the functions of DNA in living beings. Types of DNA. Three main kinds of double-stranded DNA are known to exist. These are A, B, and Z form of DNA. The most usual form of DNA present at neutral pH and physiological salt concentrations is B form, that is right-handed double helical assembly. A relatively denser right-handed duplex with lesser distance among the base pairs has been labeled for RNA-DNA duplexes and RNA-RNA duplexes. This is known as A form nucleic acid. In contrast to right-handed A and B DNA, Z form of duplex DNA has extremely diverse left-handed helical assembly shaped by stretches of interchanging purines and pyrimidines. For example, GC, particularly in negatively supercoiled DNA. Only a small proportion of cellular DNA exists in the Z form. It will be interesting to correlate that different structure is involved in some way in regulation of some cellular functions like transcription or regulation, but convincing evidence for or in contradiction, this proposal is not accessible yet. Important differences between these three different types of DNA are, for example, regarding the direction of helix, A is right-handed, B is right-handed, but Z is left-handed. Regarding the shape, A DNA is broad, B DNA is intermediate, whereas Z DNA is narrow. Diameter of the helix, it is 2.3 in A DNA, 3.4 angstrom in B DNA, and 3.8 angstrom in Z DNA. Glycosidic bond, it is anti in A, anti in B, whereas alternate anti and syn in Z DNA. Number of bases per turn 11 in A DNA, 10.4 in B DNA, and 12 in Z DNA. The pitch size is 25.3 angstrom in A DNA, 35.4 in B DNA, and 45.6 in Z DNA. Tilt from axis is at 19 degree in A DNA, 1 in B DNA, and 9 in Z DNA. Major groove is narrow and deep in A DNA, wide and deep in B DNA, whereas it is flat in Z DNA. And minor groove, it is wide and shallow in A DNA, narrow and deep in B DNA, and narrow and deep in Z DNA. Regarding the occurrence, A DNA mostly occurs in dehydrated conditions, B DNA in hydrated conditions, whereas Z DNA occurs in dehydrated high salt concentrations. Syn and anti implies to alignment of the N-glycosidic linkages between deoxyribose and nitrogenous base. In anti orientation, base extend away from the deoxyribose, whereas in syn orientation, it is above the deoxyribose. Pyrimidine can only be in anti orientation, whereas purines can be in either of the two. DNA supercoiling. Supercoiling of DNA was discovered by Vinograd and co-workers in 1963. The coiling of DNA could be of two types. First, positive coiling, when DNA molecules is coiled in a direction similar to the direction of twisting of the two polynucleotide strands tend to overwind. This type is called positive supercoiling and is formed in eukaryotic chromosomes. Second, negative supercoiling, when DNA molecule is coiled in a direction opposite to the direction of twisting of the duplex the two strands tend to unwind. This type of coiling is known as negative supercoiling. It is found in circular DNA present in bacterial viruses and mitochondria. Now let us summarize what we have learned in this module. DNA is a biopolymer that contains and encrypts the genetic information applied in the growth and functioning of living systems. DNA, together with proteins and carbohydrates, is one of the three chief macromolecules. DNA is made up of two antiparallel strands that are coiled around each other to form a double helix. The strands of DNA comprise of simpler units known as nucleotides. Nucleotides are comprised of a nitrogenous base, purines, guanine, adenine, or pyrimidines, thymine, cytosine, as well as a monosaccharide sugar and known as deoxyribose and a phosphate group. Nucleotides in DNA are linked to one another by phosphodiester covalent bonds among the sugar of one nucleotide and the phosphate of the subsequent, resulting in a sugar-phosphate backbone. The purines forms hydrogen bonds to pyrimidines, with adenine forms bonding with thymine by two hydrogen bonds, and cytosine bonding only to guanine via three hydrogen bonds. A set of purine pyrimidine nucleotides bonded together around the double helix is called as base pair. DNA is a highly stable molecule, making it suitable to store the biological information.